uracil and capacitabine, they have a big problem that is neuropathy, something called the Hanfield syndrome, and that's a very, very big problem. We know, we have been extensively working in trying to identify biomarkers to predict secondary effects, and some of them have been found, and some of them are nowadays have to be used in clinical practice because, at least in Europe and the States, it's mandatory because they have been included in the technical sheet by the regulatory agencies. In the case of the States, the agency is the FDA. In the case of Europe, the agency is the EMA, the European Medicine Agency. For instance, there is a variation in a metabolic enzyme, the UGT1A1, that the people having this genotype, they have a problem that they have that is the main cause of neutropenia for when irinotecan is used in combination or as a single agent. And this is a technical sheet of the irinotecan by the FDA, and they say that when this drug is administered, it should be considered to reduce the doses in patients that they are homozygous for this allele. But they say, however, the precise dose reduction in this patient population is not known, and the subsequent dose modifications should be considered on individual patient tolerance, based on individual patient tolerance to treatment. We have been also working on that, and now we are able to predict and to calculate, depending on the genotype, the dose reduction that we have to apply in order in people having these alleles, these genotypes, in order to make a safe treatment. But the question is to look for new biomarkers, because still the adversary reactions are terrible. And for this, we don't have any other choice than to apply association studies as well. And there's a big difference using association studies for looking for genes involved in diseases than for pharmacogenetics. Candidate gene approaches that have been a complete disaster when we look for genes involved in complex disease work much better in pharmacogenetics. And the reason is that we know much better the metabolism and the transporters and the targets. We understand much better the drugs than the disease itself. Second, when we are looking for adversary reactions, we need only very few cases and controls. Look at this case, for instance, that is a paper published by the group of Endale in the UK, that to find an extremely good biomarker to predict the simvastatin-induced muscle toxicity, they have used only 85 cases and 90 controls. The reason is that the phenotype is much, much better defined. And the problem is that for adversary reactions, sometimes we also need to organize consortiums, because sometimes they are very, very rare events, and we need to collect information from everywhere. Using candidate gene approaches, we have been able to identify new biomarkers. For instance, in this gene, the DPYD, predict toxicity for a number of effects, especially neutropenia, for capacitabine and 5-fluorouracil-related toxicity, using only 400 cases and analyzing 85 candidate genes. And of course, GWAS analysis are also key to identify biomarkers for ADR prediction. Last year, we made a GWAS in a little bit more than 300 patients from Spain that we have very good pharmacogenetic data of adversary reactions. And we'll replicate this in a population in samples from the UK. And these are the secondary effects, and these are all the SNPs. And we were able to identify all these SNPs related with a number of the ADRs. This one related with mucositis, that it has an incredible odds ratio value, is going to be included in the technical sheet of the 5-fluorouracil by the European Medicine Agency, and by the end of this year, probably. When in a second-line treatment, when the colorectal carcinoma has metastasis, there is a big improvement in treatment with the use of what we call the anti-EGFR antibodies. This is a complete revolution in the treatment of metastatic colorectal carcinoma. The drugs are called panitumumab and rituximab, and these drugs block AGFR, and in this way avoiding proliferation and metastasis and angiogenesis. But the problem is that when the KRAS gene is mutated, it's not efficient because it's permanently switched on the pathway, and then blocking EGFR will not prevent events downstream of KRAS. And around 50% of the patients have this in the tissue have wild-type tumors, but the other 50% is mutated. And there were many clinical trials demonstrating this, and not only demonstrating the efficacy, but demonstrating that people having mutations in this gene, this therapy is inefficient. And the health authorities, I mean the FDA and the European Medicine Agency, have indicated that patients having mutations in the KRAS codons 12 and 13 should not receive cetuximab or panitumumab. And there are different mutations. But the problem is that it's not so clear to apply this in clinical practice, because first, the KRAS analysis depends on the sensitivity of the technology. So this is not so clear. Second, not all the mutations have the same value. There are mutations in these codons that can benefit of these therapies. There are many mutations in other genes that we don't know the degree of resistance, and this is mainly unknown. So there is still a lot of research that has to be done. And perhaps the most interesting advance from my point of view in this subject is there were two papers published in Nature last year that by two groups, that they found that mutations, obviously in KRAS, are associated with that curve resistance to anti-EGFR treatment. But they have been able to apply methodologies, very sensitive methodologies, using biomodalical PCR combined with flow cytometry technology and with next generation sequencing technology, and analyzing DNA in blood. They found, analyzing tumor DNA in blood, they were able to find mutations and to predict the outcome and months before following treatment. And that's very, very important, because that means that the KRAS mutant clones can be detected in an non-invasive way before radiographic progression. And most important, these resistant cells remain sensitive to the combination of AGFR and NEC, and that's a very promising finding for future therapies and identification of this type of people. But still, there is always a long way to go from research to clinical application. from lab to bench, from bench to bed. There is always a long distance. But and I think the same as I think that the future is obviously integrate things. I have been talking because I have only 25 minutes, and I have been always about genomics and approaches to identify pharmacogenomic biomarkers using genomics. But it's not only structural things in DNA. We have to talk about microRNA, transcriptomics, epigenomics, metabolomics, all the omics together in a common framework of integration and interpretation is obviously the future. But at least for some of them, the next generation sequencing technologies allows us an opportunity to integrate some of these technologies together. And I think that is going to be the next step. Well, so in summary, I would like to send this message. First, biomarkers for adversary drug reaction prediction in first-line chemotherapy for colorectal carcinoma are being discovered thanks to the use of association study with both candidate genes and GWAS approaches. Second, this type of studies yields more results for pharmacogenomics than for common disease susceptibility genes due to a better definition of the phenotype and a better knowledge of the pharmacogenomics pathways. Third, the use of a comprehensive panel of biomarkers produces response and some secondary effects, and it can be efficiently translated to clinical practice. We are using this nowadays every day in our hospital and in many hospitals in Europe. But more studies and new biomarkers are needed to improve prediction. For anti-EGFR treatments with monoclonal antibody, KRAS mutations are usually analyzed in tumors, but still studies and standards are needed. And the molecular evolution of acquired resistance to targeted EGFR blockade in colorectal carcinoma needs still to be understood. And next generation sequencing, combined with other high sensitivity approach and probably in tumor DNA in blood, are needed to be investigated to make progresses and to integrate information. And that's all. For me, it's important to thank all the group, all my group, that is this. And all the consortiums where we are involved. Because in this, genomics now is a very collaborative science. And it needs to work all together. Clinicians, geneticists, people dealing with new technologies, mathematicians, and we need to organize big consortiums to collect samples in the same way. In our case, we are involved in the EPICOLOM consortium, in the COGEN consortium, in the CHIPCHA consortium. This is a consortium paid by the European Union for Latin America. The DELIS consortium is a consortium from the state, sponsored by the NCI, the NIH in the States. And here I think we are doing also, I think that we are going to have very good projects in this regard. And one of the projects that we have proposed has been approved, and Mahmoud Rasool is the PI of the project. So I think we are going to find, with all these efforts, new biomarkers. And because everybody is always asking me, where is Santiago? Because most of the people only know from Spain, Barcelona, and Madrid. Here we are, in a part of Spain that is very old. This is Santiago. It's a very old town, mainly built in the 11th century. It's a pilgrim place, the same as Jeddah to some extent. We live in a part called the Finisterre, this part of Spain was called by the Romans the end of the world, because it's the westernmost point in continental Europe. So we are far away from everywhere. But despite of that, I am very happy living here, and especially I am very happy because I have a very good group with many people, because we are doing clinical case work and research at the same time. So we are always interested in translation, and I am very happy with the people I have. And in my group, you are all very welcome. And I am this one, that I think I have to finish my talk.
very much, Angel, for the great talk and insight into both discovery and treatment. Questions? Okay, since it's taking some time, let me ask a couple of very short questions. I noticed in your analysis of the transcriptome, you didn't have an arrow pointing to functional characterization of the mutation. So you think that the transcriptome is not going to be useful in functionally characterizing individual variants?
Yes.
So there's a pink arrow missing pointing upwards.
In general, that's a key question, a very important question. For a biomarker to be approved by the agencies, the regulatory agencies that we have, the functionality has to be always proved.
Mm-hmm.
That's the first thing. Otherwise, so we have always to make functional studies for the biomarkers that we are identifying. The same is true when we are trying to identify a gene involved in the disease. We can identify the genes or the region using a GWAS approach, but then we have to make functional studies. I think the paper that you have, this paper that probably you are going to talk about is in this correlation between the transcriptome and the structural variation in the genome that you have coordinated in this consortium that we have published in Nature a couple of months ago. Mm-hmm. Is going to be key to try to compile structural variation with functional variation. I think that's the way to go ahead. And but anyway, anytime that we identify a gene or a marker, we need to make always functional studies to try to prove what is happening behind.
Mm-hmm. Any questions? Who's having the microphone? I know.
I have a question, please.
Okay, yes.
Thank you, Professor, for such a great presentation. I just wondered if what's happening in colorectal cancer will also be needed to be applied to other tumours, or is it once you give a drug to a person, then you can associate certain markers that will tell you the effect? Is it a general effect or is it a tumour effect?
She's asking, well, probably you have the same problem with me. She's asking whether it's the same trend for other cancers beyond just colorectal cancer.
Of course, everything is exactly the same. I mean, this can be applied to any other cancer. And most of the cancers are exactly the same. Most of them have more or less the same heritability. The same is true for everything. And I think that one of the things that I forgot to mention is that perhaps one of the most important questions for the future of finding biomarkers for treatments is going to be the sequence of tumor stem cells. That's an approach that is going to be key to understand the drug response and to identify different groups. And this is a project here also ongoing on neuro-oncology, I mean in brain cancer. I think that is going to be key to understand not only the molecular causes of the tumor and to substratify the tumor, but also to understand drug response is going to be key. But the same I have mentioned here for colorectal carcinoma can be applied to any other cancer.
Okay, there's one more question out here.
Yes, thank you very much for this nice talk. Actually, we have a group of patients. We found that they have 11 novel variants. It hasn't been reported yet, but my question for this one, have you seen any variation in terms of the severity of the disease and response to the treatment for other biomarker rather than RAS gene?
I think the question was, have you seen any variation in the severity and response to treatment other than the RAS gene? Is that correct?
Yeah, I mean, when you see these variation, I mean, the SNP variation, is there any correlation between type of SNPs and the severity of the disease in terms of the severity and the response to the treatment? If you add it to the RAS factor as well as treatment. Thank you.
Yes, when we use this, we have identified many SNPs using this GWAS study. And we used to make always a correlation. Once we identify the SNP, the next step is to find sequencing to try to identify the gene, and then to try to provide functional evidence for this variation. But one thing that we're always doing is to try to establish genotype-phenotype correlation. And some of these SNPs and some of the combination of the SNPs, they are correlated also with the phenotype. And some SNPs and some variations, especially some combinations of SNP, they are related with bad prognosis, and they are related with some phenotypic characteristics of the tumor. And we are trying to understand this a little bit more. At the end of the day, for this type of variation, gene-gene interactions and gene-gene environmental interactions are going to be key to understand the different phenotypes of the disease. So we are trying always to correlate this, and in fact it exists, a correlation between the severity of the symptoms in some cases and some combinations of SNPs.
Okay, well, we have to move on. Thank you very much, Angel.
Thank you, Manolis.
Okay, so we're continuing with colorectal cancer, and we move on to Dr. Abdelbaseb Boumeida, who's going to talk about molecular characterization of colorectal carcinoma of Saudi Arabian patients and identification of predictors of disease outcome.
Thank you very much. Thanks. I'd like to thank the scientific committee for giving me this opportunity to share with you some results from our colorectal cancer, Saudi colorectal cancer, in our genomic center. As we know that colorectal cancer is a complex disease and it's a heterogeneous disease. And according to Saudi Cancer Registry 2009, it is the most common disease in men, Saudi men, and third in women. And there is increasing and the incidence rate between 2007 and 2009, which means that increasing progressively. This increase could be due to the change of the lifestyle, diet, and physical activity. Still, we don't know exactly, but we are doing our effort to get more at molecular level. And because it is a complex, many people, scientists, consider it as a disease hits two organs, because we have left colon, right colon, rectum, and these are at molecular level different, acts as different organs. This heterogeneity is mainly combined due to multitude of genetic and epigenetic variations acting as messengers or drivers. In general, for normal cell to move from normalcy to malignancy, events should be happen or mutations at oncogenes or tumor suppressor genes. So oncogenes should be activated and tumor suppressor genes to be inactivated. Tumor suppressor genes inactivated either by loss of activity due to mutational or deletion, or a change at epigenetic level, either DNA or maybe microRNA or histone. So these modifications leads to the inhibition of the gene and leads to inhibition of tumor suppressor genes and leads to cancer. In colorectal cancer, the majority develop via chromosomal instability, which this instability exacerbated by activation of wind signals, ABC gene, mainly, or mutations of KRAS, BRAF, or deletions of P53, etc. Also, mismatched repair genes, it's a set of genes very important for integrity of the genome. If there is any defect of them, it could lead to malignancy, mainly MLH1 and MSH1, MSH2 gene. For methylation, CpG line SEM is very important pathway for sporadic colorectal cancer. However, so due to the heterogeneity of the colorectal cancer, the traditional pathological grading system and staging system, although they are powerful as a prognosticators for the treatment, still inadequate to predict the outcome of the patient within the same stage. So, stage 2 patients will behave differently. If we have 100 patients stage 2, they behave differently. So, we are in need of molecular markers to pick up those patients with high recurrence, to go to personalized scale. Therefore, an active research is going for finding powerful prognostic and predictive tools to identify high-risk patients who would benefit from tailored treatment. So my presentation, just to summarize a role of some molecular markers, we studied them in our center, and to pick up those that have potential as a predictors to extend our studies in the future for precise medicine. So in my talk, the patients and method available, we use methylite assay for methylation profiling, epigenetic profiling of our patients of some candidate genes, and also we use immunohistochemistry for specific proteins to see the expression of these proteins and if they have a prognostic potential. In this talk, we use just disease-free survival and disease-specific survival as a monitoring for the patient outcome. And for methylite, so these are the candidate genes, 15 genes. We scan them for methylation profile, and the other for proteins by using immunohistochemistry, we use MMPs. Also, we study some of the cell adhesion molecules, cell cycle regulators, COX, and others are under investigators, like family of fibroblast growth factors, clothers, and some of biological clock genes like PMR and melatonin. These are under investigation. Also, we study MSI status by using 5 markers for MSI. This paper, just we study the epigenetic or methylation of these candidate genes by using methylite assay, and these are the primers and the probes of these candidate genes. And in this study of this methylation of 15 genes, the methylation profile is shown in this table, that these are the black, white, and gray is the side of the tumor, right, left colon or rectum, and here's the methylation profile. And these polycomb, which is very important, they are stem cell genes are methylated stem cell. These are polycomb group target genes are highly methylated in our group, and SIM markers, also 5 markers, is a little bit low methylated in our samples cohort, and mismatched repair genes, the lowest methylation. And this methylation profile we extract and to find if it's act as a prognosticators, which is one is very interesting, that the Vericom BCG genes, SLIT, FRB1, MyoD, and HIC, they are mainly methylated in the tumor as comparing to the normal, matched normal. This is the matched normal, and here's the tumor is highly methylated in the tumor as compared to the matched normal. The overall average of methylation of our samples comparing to the worldwide average of these the same genes, some of the genes is hypermethylated in our samples compared to others, and others are hypermethylated in worldwide through the PubMed.org database, and but the overall methylation is almost the same between our samples and the worldwide samples. Very interestingly, that through using clustering analysis, we distinguish 3 groups by through the methylation of these genes, and we found that group 1 is a methylation low, numbers 36, group 2 methylation intermediate, number 67, and group 3 methylation high. Through these methylation, we found that group 2 behave better than the others. So this is the clustering. Here's the group 1, low methylation, group 2, intermediate methylation, and high group 3, high methylation. The black dots are hypermethylation, the gray is non-methylated. And through this, by using survival analysis, we found that group 2 behave better than the others, and group 2 possesses the worst prognosis. Also, we did mutation analysis of KRAS in our cohort, 120 samples. It's lower than worldwide. 20% we have KRAS mutation and 2.5 BRAF mutation. And for the MSI status, 45% our samples are MSI high and 24% are MSI stable. And also we use comparing the MSI of 2 genes, MLH1 and MSH2, the concordance of these by PCR and immunohistochemistry, and we found that the concordance is between the loss or the mutation and the loss of expression is 60% concordance, and 5% is a concordance, and the non-concordance maybe due to that we didn't look for the other other proteins mismatched like MLS6 and BMS2. So these are under investigation. I would like to see in the future what they wrote in our samples. This is the immunohistochemistry just to show loss of MLH1 exhibition. This is a normal exhibition and tumor lost. Here is the positive MLH1, that normal positive, and also cancer is positive, that's not lost, and here is lost. The other is for the MSH2, which is very important, mismatch repair gene. Here is normal expressed and tumor not expressed, and also here positive means both normal and tumor expressing the protein. Other work that we just see the impact of B21 and B27 as the prognosticators of our samples, and it seems that this by immunohistochemistry that B27 is more powerful than B21 as prognosticators. The other marker cyclooxygenase 2, which is very important as a predictor in our samples, and the work of cyclooxygenase 2 act through by remove or act on free arachidonic acid to make prostaglandin E2, which losses tumorigenicity, proliferation, and angiogenesis. So high exhibition of COX-2 is bad prognosis, and this is the by immunohistochemistry. This is the exhibition of the COX-2, and here's already differentiated with lost, and these stromal and lymphocytes lost, while the tumor is negative, and this survival showing that positive cyclooxygenase has a bad prognosis compared to the negative. Also, we studied vilin as a protein to see if it has any impact as prognosticators, and it's 100% expressed in normal colonic mucosa, while 84% or 85% of our samples showing the positive vilin, and this is the by immunohistochemistry normal and well differentiated intermediate, and this is already differentiated loss of vilin, and vilin seems that high vilin behave better than negative or no expression of vilin. Also, we study CD44, and it seems that CD44 has no prognostic impact on our samples, and we would like to extend this to prove it in more conclusive result. Also, we studied some matrix metalloproteinases, a family of 16 proteins, but we selected MMP2, MMP7, 9, and 10, and see if they have impact in our samples. And this is the expression by immunohistochemistry. And through survival curves, this is the disease-free survival of MMP2. So patients with high MMP2 behave worse than low MMP2. Here is the disease-free survival for MMP9, and here is the disease patient with high MMP9 and high MMP2 die of cancer than patients with low expression. And we make a combinatorial approach that's lumping MMP2 with MMP9. We found that positive MMP9 and MMP2 has the worst prognosis, and the best is MMP negative. If the patient with MMP negative 2 and 9 negative behave better, this is the disease-free survival. And here is the combinatorial approach for those two proteins, and showing that MMP9 here as a disease-specific prognosticator is more powerful than MMP2, but both, if they are negative, patients live better than the others. Also, we studied cell adhesion molecules, selected E-cadherin, beta-catenin, alpha-catenin, and cadherin. This is very important for the homeostasis and for the coherency of the epithelial cells. This is the expression of E-cadherin, membranous or cytoplasmic, beta-catenin, also nuclear and membranous cytoplasmic and cytoplasmic, alpha-catenin if positive, and N-cadherin if it's positive or negative. Those, and we see that alpha-catenin, patients with high negative alpha-catenin is bad prognosis, with positive high expression is better. And for the N-cadherin, the positive N-cadherin worse than negative N-cadherin. So, but we didn't find any correlation with E-cadherin and beta-catenin, unfortunately, with our samples. And before I finish with my talk, just to remind you that usually in the immunohistochemistry, the slides, when stained and scored by expert people, they discard or put in an archive and not use it. But we think why not to reuse the already stained immunohistochemistry slide to crude dissect the tissue after immunostaining and to extract the DNA and to find if the integrity of the DNA is okay and can be used for further analysis. And we just designed this protocol that we we put the already stained slide in xylene for 1 week and then crude dissect and extract the DNA for further methylation analysis or mutation analysis. And it seems that we succeed to take an example, for example, CDH1, which is E-cadherin, that this is E-cadherin through protein expression immunohistochemistry, and here is the methylation, and found that the concordance with hypermethylation and loss of the protein or expression of the protein, and there is low expression. The concordance rate is 60%, and it's almost the same as in the literature. This is epigenetic, and also we test the DNA integrity for the beta-catenin CTNB1 gene, and this is the electrogram. It seems that it's okay, and would like also in the future to see if we can extract RNA from that already stained cells for further molecular studies. So if I can conclude that understanding genetic and epigenetic alteration as driving forces cancer open new field of research, epidemiology, risk assessment, prognosis, and treatment. Colorectal cancer provide a good model to explore the molecular basis of carcinogenesis. Mismatched repair genes was rare in our cohort, and the contribution of inactivation of mismatched repair genes in Saudi population is yet not to be known, still not well explored, at least in our region. The KRAS mutation and BRAF, it seems that they are lower than what's registered in the worldwide, maybe due to ethnic differences. SIM frequency is almost 14%, was similar to the previous reports. Now our samples group 2, BCG2 methylator and SIM negative signature carried favorable prognosis, and the methylation of BCG, RASF1, ABC, MGMT for repair gene could be used as a new biomarker for classifying colorectal cancer. B27, COX2, Villin, MMP2, 9, catenin, alpha, and cadherin all have prognostic potential in Saudi colorectal cancer, and MMP2 and 9, alpha catenin, they retain their independence. They are independent of the stage and other molecular markers, while B21, E-cadherin, beta-catenin, CD4 were less or have no prognostic impact in our colorectal cancer cohort. Unfortunately, the sample size of these cohort studies is too small to allow for more definitively, because the number is 120 samples, and this make our for that to increase the work in the future. Our future plan just would like to study gene expression, and this should be the first study, but due to the logistic delay, we are waiting for the ships, and we have almost fresh samples, 65 samples. All of them passed the quality control and ready to be studied by gene expression with matched normal. And also, we would like now in the process of building a tissue microarray database slice of 300 samples, and this is a block. This is a new this package of 300 colorectal cancer with full pathological and clinical data to be constructed, and we are in the middle of that for validation of different biomarkers in the future. So I'd like to recommend that establish full colorectal database with complete follow-up data to facilitate further research work. More large cohort studies are needed to evaluate the role of different molecular markers as prognosticators. Scientific collaborations at national and international level are highly recommended for transferring technology, exchanging research ideas, and also to compare our results of Saudi patients with other populations. Establishment of the infrastructure, prognostic and predictive genomic models with molecular signatures is very important to facilitate the approach towards personalized oncology in the Saudi health system, and we hope that in the near future we reach to this level. Finally, I would like to acknowledge Ministry of Education, CAPS, King Abdulaziz Institute for Science and Technology, Colorectal Cancer Scientific Chair of King Abdulaziz University for their financial support, CGMR staff for their nice work and collaboration, and also national and international collaborators for logistic help and logistic support. Thank you.
Okay, thank you very much. We have time for one short question. Down here, up there, any question?
Please, no question.
I will ask one. So do you think that the differences that you've observed so far in the Saudi population, even with a limited sample size, justify an in-depth analysis of the Saudi population?
No.
Or just the transfer of whatever is found elsewhere is as good?
Yes, exactly. So this is just the preliminary studies to show the molecular heterogeneity of our samples. And to be honest, this is only candidate genes, limited genes, and we would like to explore at transcriptomic level and gene expression, which I think that better to start with them to pinpoint the list of genes that are very important for our. But this is just to comparing and to get used with the technique and to compare with other worldwide molecular characterization of colorectal cancer. But to be honest, this is still limited number and cannot give a conclusive output.
Okay. Well, thank you very much then. What? Oh, there's one more question. Okay, go ahead. Short question, please.
Thank you, Doctor, for your presentation. One of the table in the presentation regarding the BRAF mutation, some patients with colorectal cancer, they have a mutation of BRAF gene. And this is well known for us. Also, it's the mutation also for patients with melanoma, for patients with thyroid cancer. Last year, we have a medication targeting this mutation, BRAF mutation, imurafenib and darbepoetin. This is already existed last year and approved by FDA. Also, it's approved for melanoma cancer with BRAF mutation. Do you think if we use this medication, this molecule that targeted BRAF, it would be get a benefit or it would be effective for those patients?
I think, but in our samples, just 2.5%, not in our cohort, 120.
Even that, that's a personalized medicine.
Yes, yes.
Thank you.
Okay, thank you very much. Moving on to Dr. Brian Mayer on application of next-generation sequencing in oncology?
That the number of genes reported with respect to inherited diseases has increased dramatically over the last 2 years. I think this early success in next-generation sequencing has then prompted agencies such as the International Consortium for the ICGC, for instance, the International Cancer Genome Consortium, and things like the Cancer Genome Anatomy Project to use next-generation sequencing as a means to develop biomarkers for cancer. Clearly, I think from talks this morning on the use of metabolomics for identifying markers, once again, the point was made that if you search the literature for cancer and for biomarkers, that you get an enormous number of hits. But yet, I believe that these hits are not translating into cancer as rapidly as one may wish. The reasons for that, though, are quite obvious, that the identification of markers is one thing, but then translating them into the clinic often takes a lot more work. However, it's very encouraging that clinical trial design is now stratifying these tumor-based markers, stratifying into their studies these tumor-based markers with a view to trying to identify predictive markers that will tell you which drug is most likely to give the best outcome for a tumor, prognostic markers, how is that tumor going to behave, or pharmacogenomic markers, as we had in an earlier presentation today. When you look at next-generation sequencing as applied in cancer, essentially the technology is fully leveraged, where one is looking at sequencing at the whole genome level, which you can see here, where essentially a genome is fragmented and sequenced in its entirety. However, even in the large consortia, it is becoming evident that the amount of work and the amount of data proves to be a challenge when dealing with whole genomes. This, to some extent, is being circumvented by looking at whole exomes, and I think a large number of the cancer studies currently undertaken actually look at whole exome sequencing. Whole exome sequencing relies on creating exon baits to pull out the exomic sequences. This represents probably somewhere in the vicinity of 1 to 3% of the genome, a much more manageable amount of data, and one would argue perhaps the most important data within the genome. There are also amplification-based technologies to target specific areas such as the exome, and transcriptome-based sequencing, either the transcriptome in its entirety or once again selecting particular areas of interest for sequencing. There are many advantages for detecting mutations with next-generation sequencing, the first of which is the high throughput that is offered by these technologies. It allows you to test many genes at once, or indeed whole genomes or whole exomes. However, in terms of testing many genes at once, there are now many cancer panels available that are being used through different modalities of next-generation sequencing, and probably offers the first opportunity to translate a lot of the findings from next-generation sequencing into a clinical or a diagnostic situation. Next-generation sequencing actually offers a systematic and unbiased means of detecting mutations. It detects all mutation types, from single nucleotide variants to copy number variants, including alterations, insertions, deletions, and given the right library construction, also the ability to identify novel translocations that may be present in tumor cells. The digital readout of this technology and the high throughput also gives you an opportunity to identify rare events, particularly important in cancer, where you have a very heterogeneous sample, meaning that the initial tumor may be a mixture of both normal cells and also be a mixture of multiple clones within a single tumor. It does offer a cost-effective approach which can lead to precision or personalized medicine, which simply put is the right drug at the right dose to the right patient at the right time. However, next-generation sequencing also has many unique challenges in its implementation for cancer diagnostics. The first of which is the complexity of the cancer genome itself. Cancer genomes are extremely complex and diverse. There are multiple mutations that occur within the tumor. The degree of variation in cancer cells compared to a reference genome is enormous. If you're looking at a simple inherited disease, essentially you're looking for a single change relative to a reference sequence. However, in cancer, given the genetic instability of that disease, there are many, many mutations that are to be identified and validated against a reference sequence. Also, most tumors are aneuploid, which is to sort of say that they have increases or decreases in their genetic material, whereas much of the bioinformatic structure associated with next-generation sequencing, currently at least, addresses a diploid status. The genome structure of cancer cells also proves to be challenging in that there are many translocations present which require differences or more complexity in terms of library construction and places more pressure on the technology with respect to things like read length in order to give you the ability to identify novel translocations which could lead to, for instance, fusion genes which may be drivers for the particular cancer and potential targets for that cancer. Also, the genomic alterations in cancer can be found at low frequency. That is to sort of say that subclones may actually represent a minority in terms of the variance within a sample, and therefore, in order to be able to detect this, you need a technology that has a very high throughput that gives you a large depth of read to be able to identify alterations present at low frequency. The samples may also vary in quantity, quality, and purity from a constitutional sample. In a simple genetic disease, it's very easy to take a blood sample to extract that DNA and to know that you have good quality material to proceed with next-generation sequencing. However, when dealing with tumors, often one may be faced with a limited biopsy sample or archival material. When you're working with limited biopsy material, the use of whole genome amplification, for instance, is not ideal, as this would affect things like the incidence or frequency of rare mutations within the sample, or certainly bias some of the questions that one might want to ask with respect to copy number variants. The quality of samples, often when dealing with cancer, people reach into the archive and use things like formalin fixed material, where DNA can be fragmented. It's not an ideal sample to use. If you're using fresh samples or even archive material from tumor, the presence of either necrosis or apoptosis in that tumor will once again affect the quality of libraries that one can build for next-generation sequencing. The tumor heterogeneity, I've already sort of touched on that, in that tumors will have both a mixture of normal cells and multiple clones, and to be able to identify rare variants, for instance, in a stem cell population of tumor, could cause problems if that cell type is actually infrequent in the whole tumor. So, when one looks at sample procurement, pre-analytical issues, even with things like formalin fixed tissues, there are still issues with the age of these samples. Often, I think people are working with samples that have been archived for decades, and not simply for years. The way those samples are being stored and processed can often affect the type of results one gets and cause difficulties in terms of interpreting the changes that one might find when processing these through next-generation sequencing. Fresh tissues, whilst at first one might consider to be ideal, are not necessarily so with tumour, because you will be working blind with respect to what the structure of that tissue is. Just what percentage of that tissue is tumour, what percentage is normal, are you dealing with the right piece of tissue? These questions come up when dealing with fresh tissues. With fine needle biopsies, where one may be able to get a fairly pure tumour sample, the small cell numbers, again, cause difficulties with being able to apply this technology. NGS methods really already use very small amounts of DNA, down to the nanogram levels, but when you're talking about dealing with a few cells, one then needs to develop methods that can create libraries from picograms rather than nanograms of DNA. So what does all this mean with respect to implementation of NGS for clinical cancer diagnostics? I think in terms of clinical next-generation sequencing in cancer, the goals are to have a high throughput technology that is cost-effective and can give you deep coverage. Deep coverage being necessary to be able to identify these minor clones that may be present within the tumour sample. You also want to be able to target clinically actionable regions in a clinically relevant time. So it's not always going to be possible to get a tumour sample and to be able to do a whole genome or a whole exome in a time frame that is useful, either diagnostically or with respect to monitoring the clinical course of a patient. This will be particularly so if you want to be dealing with not just single samples or tens of samples or hundreds of samples, but thousands of samples, as one would predict with the development of personalized medicine, or moving into the time where you would envisage sequencing every new tumor that presented in the clinic. So this is particularly where I think the sequencing of panels of genes, now rather than looking at a whole exome that may cover some 25 to 30,000 genes, being able to focus on a set of several hundred genes, I think offers the immediate application of NGS with respect to the clinic. Even at that level, there remains challenges, and one of the challenges is the huge infrastructure costs associated with putting in the technology to do the sequencing